Thank you for everyone for being here. It's great to be here myself. Um, so Nano Day is coming up soon, and I thought we should make this a little bit more of a fun experience. Maybe if this is recorded, people can learn more about nanotechnology going forward. So I thought I'd start with a little story. I was recently visiting some of my family in the great state of Kansas, and not to boast, but I have a pretty family. They have a lot of great accomplishments that I admire so much. But none of them are researchers. And so, they're asking me, like, Abigail, what do you do in nanotechnology? What is the Cancer Research Institute doing on a day-to-day basis? What is this like? And so I was like, okay, we have hundreds of people here. I need to figure out a way to talk about this and convey this in as few words as possible, and get the meaning across of just how valuable this community is. So I said, I think everyone knows who this guy is. This guy is James Bond. And they said, yeah, okay, we know who James Bond is. And said, okay, good, we're on a good starting point. I was like, this is who I work with in the Cancer Institute. I work with the James Bonds of the cancer criming family, where you are all going into the depths and really understanding the root of the foundation of the criminals in the cancer world. And they said, okay, yeah, that makes sense, but what do you do? It's like, okay, well, James Bond is, we all agree, and he gets an awful lot of support from this guy. And for anyone who hasn't seen these movies, this is Q. Q works in a top secret lab where they're developing really exciting technology. He will give Bond on his debut of his mission a fancy pen that only shows up when you splash a martini on it. And by the way, it can also, explode into a parachute if they think he's falling off a building. So it's like, that's what we try to do. That's what we try to bring to the cancer research community. And so my family said, okay, yeah, that makes sense. You're basically using nanotechnology to pack a parachute into a pen to solve cancer research. I was like, yeah, I think that sums it up. Does everyone feel like that describes what we do here? Just give a quick overview of sort of the breadth of capabilities that we have. So if anything sparks your interest, you can come down and talk to us and learn about it in more detail. I think a good way of describing us is not as a single technology laboratory, but more of a landscape of technologies where we have many what may seem disparate technologies, but that are all focused on a similar goal of imaging and nanomaterials work. So the instruments that we have up here, these are really accessible instruments. These are instruments that you can come down, we can train you really quickly, and then you can learn how to use these instruments and operate independently. So these are instruments that allow nanoparticle characterization. This instrument, for example, you can, in a high throughput format, you can screen hundreds and hundreds of molecules or particles very easily to look at particle sizing information. We have another instrument that is similar in that it measures nanoparticle size, but it does so at a much higher resolution. So for example, if you had a mixture of different populations of particles, you could differentiate these individual particles, and it also gives you concentration data in particles per mil on the y-axis, which is a really tasty bit of information to have if you are trying to do downstream applications. Interestingly, like if you took this sample and you ran it on a DLS instrument, you would generate what you see in this green curve here. So these different technologies generate very different data, and it's important to understand where the differences come from. If you are a hydrogel worker, perhaps you're designing hydrogel materials, incorporating nanoparticles, different types of drug delivery. You may be interested in using the rheometer for understanding modulus and viscosity. And we also have an AFM for atomic force microscopy. This is an example of some DNA origami that was imaged on this microscope. So the instruments that you see down here, these are our 4 different imaging instruments. And there's a selection of different SEM, TEM electron microscopes, both cryo and room temperature. And we also have a confocal that can work in cryo. It's sort of it's super resolution curious. It dips its toe into the super resolution world. And it's really these instruments here where we operate more as an innovation platform, where I have a team of 5 people with 120 plus years of research experience in this area. And we work collaboratively with you, project-based, to understand the work that you're doing, understand the nuances and challenges of your project, and then help do experimental design and development, workflow design and development, protocol selection and development when necessary, sample processing, transfer, handling, and then, of course, the image and data generation. And so we do this on a case-by-case, very bespoke approach. For every instrument that you see here, or not for every instrument, but there are a dozen plus other instruments that are support instruments that we use that are really what enable us to do a lot of the work, because that's most of what this kind of work requires is sample preparation. So this is just a quick, compilation of different images that have been generated in our space. I'm not going to go into what all of them are in detail, just point out like this is a cryo-TEM image of cryo nanoparticles. I mentioned a fib in our space. And this is what volume data can look like, where you are passing through cells. This includes a couple 2,000, I think, images about 10 nanometers apart. And when you look at this data in a whole, you can really see in quite detail what's happening through the entire volume of the cell in terms of the different organelles, where they are, how they're organized, changes that may have occurred to treatments that this sample underwent. So if anything here strikes your curiosity, come down, we'll talk with you and sort out what types of approaches might be best for you. But I think I'd like to spend the rest of this time, I'm just going to very briefly introduce CLEM microscopy, and then I will invite Giovanni to come up. So CLEM microscopy is an umbrella term. It's very generic. It just means any type of imaging strategy that allows the correlation of light and electron microscopy. And so if you look through the literature, you will find many, many different types of workflows. And the reason this kind of work is challenging is because often the preparation that is required for light microscopy, for example, damages the sample in such a way that you can't use it for downstream EM. And the vice versa is true. Processing for EM-based approaches often disrupts the light microscopy imaging capability. So finding ways to bring these two things together is sort of this constant challenge that we're always working on. And I thought I'd just have to ask you to do a little thought experiment where think about you have a situation, you have 2 friends. Friend number one has a superpower. Friend number one can tell if there is a criminal mastermind in the room. Now, this actually comes, unfortunately, with a caveat. This friend is blind. They can't really see structure. They can just see a glowing aura of red. And so when this friend walks in a room, this might be what they see. And they can say, oh, there is a criminal mastermind here, and they're over there. But that's it. This is light microscopy. Second friend number two also has some superpowers. They're a little bit different. This friend can see incredible detail and structure. This friend can see large, vast areas, and it can see these areas with incredible definition of very small features. But of course, this also comes with a drawback. This friend can only see black and white. And so when that friend walks into the very same room, this is what they see. Now, this is electron microscopy. They can see that there are some devious looking characters, but they can't tell you which one might be a mastermind. So I was actually realizing last night, is there anyone in the room who has not seen the movie Usual Suspects? Confess right now if you haven't, because, okay, all right, good, good. No judgment. This is a fantastic movie. And so what I need all of you who haven't seen this movie to do is right now avert your eyes, because this is the spoiler alert. So look away. If we were to use this example for Clem, what we would see is we bring the capabilities of light and EM, and then we can really say, aha, there, that is our mastermind criminal. Okay, anyone who's averted their eyes can now look back. I'd like to introduce Giovanni De Nola. He is a team member who has been with me for now 2 years, 2 years and 1 day as today, yeah. And Giovanni came to us, he did his postdoctoral work with Thomas Kirkhausen over at HMS, and then he did a few years working as a scientist in Pascal Kaiser's lab, also at HMS. And when I met Giovanni, I was really impressed by just his creative spark and curiosity. If somebody had said, oh, no, it doesn't work that way, he would go, are you sure? Let's find out. So I was really excited to find a way to bring him here to join our community. And he's been here for 2 years. And over the last year and a half, he's been developing this new CLEM protocol. And I know everyone works hard. I went through my PhD and my postdoc, and we're always surrounded by people who are working hard, but I've never actually met anyone who works harder than Giovanni. He is here at nights, he is here on the weekends, he is here on holidays, so that he can do all of this developmental work on the side while he is also maintaining and continuing projects that he's doing with this community. So this is Giovanni's first opportunity to present to this community, so please join me in giving him a warm welcome.
Okay, thank you. So let's say the common problem shared by all this CLEM technique is how to label our protein of interest. And there are basically 2 routes. The first one that is also the most used is by genetic tagging, by basically tagging our protein with a reporter fluorescent protein, and then we can follow it. And this is really useful because it allows us to do in vivo or post-fixation imaging, and is also compatible with cryo methodologies, and also is the technique that allows us the best EN morphology at the end. However, we cannot mark or label particular post-translational modification of our proteins. It's not suitable for clinical samples. And also, we may introduce some artifacts, both in the function or in the expression of this protein. And therefore, it will require validation. And also, overall, it's time and resource consuming. The other route is the one of immunohistochemistry, that since it depends on antibodies, it can detect even specific intermediates. It can label multiple macromolecules, such as nucleic acids or even lipids, and is suitable for clinical samples. However, in order to deliver our probes into the samples, we need to use detergents. And these detergents, when we analyze the sample at the EM, are going to introduce more or less heavy artifacts that will make the analysis difficult, if not impossible. So we wondered, like, is there a way to combine the best of these 2 approaches, meaning having the versatility and specificity of immunohistochemistry combined with the EM morphology that we get from genetic tagging? And our answer to this question is this technique that we invented here, 2-CLEM, that stands for Tokoyatsu correlative light electron microscopy. And we called it this way to honor the work done from Professor Kyoteru Tokoyatsu in the 70s that single-handedly laid down the foundation from this work, and is one of the most used technique until now. I'm not going to go in details on his work, but I'm just going to show you what we do with that. We take the sample, our cells, we fix it, and then we infiltrate it in sucrose before freezing it. And once frozen, we cut semi-thin cryosections. Then these cryosections are brought back in buffer at room temperature. This is what I was trying to draw here, with like a petri dish with a buffer, and then you have like your cover slip with the 1 micron section of your tissue on top. So at this point, your cell is not completely covered by the membrane, is one slice of section, or one section of a cell of 1 micron that is completely permeable to your probes, to your antibody, like a house without a roof. So we solved the problem of how to deliver the antibody or the probes to our cells without using detergent. So in this way, we can maintain the best possible morphology at the EM level, whilst using whatever antibody or probe we want. And after we've labeled our sample, we can image it either with a normal confocal microscope or even in super resolution, as we're going to see in a moment. And then the very same cell is processed and imaged under the electron microscope. And these two sets of images are then correlated and merged on top of each other so that we can see what structure was lurking underneath that fluorescence. So these slides just show some of the kind of samples we have been working with in the last year and a half. There is like different mouse tissues, human tissue, many kind of cells, even organoids, cultured neurons, dental plaque, you name it. And this technique is really, really versatile and is exceptionally useful in an environment as this one, because there are many kind of research being carried on and many kind of like needs. And for now, this technique managed to help all of this. So now with some real life example, the first one I wanted to show you comes from a collaboration we done with Professor Munchki from Dana-Farber and Professor Morelli from the Candiolo Institute in Torino, where they asked us to label in the same sample RNAs and protein. So for people that are not, let's say, very fluent in CLEM, this is something that until now it is possible, but there is not an easy way to do this. It's something that really depends and requires a lot of optimization. This comes from, let's say, a weekend kind of work. Like it was really, really easy. We took the cells. What you see here is a section of this, of a cell pellet. So yeah, this is one section of a cell pellet. And this, what you see here is the merged light microscopy with the electron microscopy image. And we are labeling in green TOM20, which is a mitochondrial protein, and in red, this RNA that we call RNA1. And they knew that this RNA1 was expressed in the mitochondria. They just looked for a visual proof of this. And so we did this double labeling, RNA and proteins. And underneath the fluorescence, that is yellow because of course it's colocalized, we always see mitochondria. And this is the perfect slide to bring up an important concept, that electron microscopy, differently from light microscopy, allows us to identify structures based on their morphology. So we know that this is a mitochondria because it looks like a mitochondria. And this is something really powerful because electron microscopy images are intrinsically rich in information. And that's why the preservation of the morphology is such an important deal. For the second example, we explored with Laurie Boyer and Paul Barthelemy the possibility of performing this storm on our TOCLAM sections. And this storm, for whoever doesn't know, is like a super resolution technique that allows us to have a resolution between 15 and 20 nanometers XY, which we are talking about 10 times better than what you saw before. And for this experiment, we took these cells. There are going to be a lot of images like this. Yeah? And we labeled for LAMP1, which is a marker that is used to detect late endosomes and lysosomes. So you can already see here from the light microscopy image that the signal is much more precise and defined. But what, in my opinion, is really astonishing is how the fluorescent signal in red colocalizes with this vesicular structure within the cell. And it shows us that these lysosomes are not empty, are full with all these kind of vesicles. And that's something that we wouldn't been able to see without CLEM. But also, I want to bring up another important point of why it's important to do EM, is that you are not only going to be able to see what's going on underneath the fluorescence, but you're going to get information also about the state of the cell. For example, if you give a quick look at the plasma membrane here, you can see that there is a lot of like endocytic activity going on. You see clathrin-coated pit, you see other kind of like endocytic vesicles. So you already know that this cell is very endocytotically active. And so you can use CLEM not only to verify your data, but also as an hypothesis generating technique to kind of like guide you for your next question and what's going to be your direction after this experiment. And this is really, really important. And this other picture, I always like to show it because it is just, in my opinion, at least astonishing how well this super resolution data correlates with the shape of this vesicular structure. Now you can tell that this vesicle is a lysosome, there is no doubt, because you have the morphological confirmation and the fluorescence that tells you. Plus, you see like beautiful Golgi, a nuclear pore, the nuclear envelope, like everything is there for you to take. Then some more super resolution. We started working with Johannes Stein, who's now a new PI in the Max Planck Institute in Berlin. And he's an expert in DNA paint, which is a super resolution technique that allows you to go even further, like sub 10 nanometer in XY, namely 3 to 7 nanometer, but also allows you to do multiplexing, meaning you can look up to 12 or 30, like let's say a couple of dozens of epitopes in the same sample. And this means that you can combine the information from 12 plus epitopes plus the morphological information of the EM. And this that we're working on right now is going to be a major step forward in the field. And also what you're going to see is the first time that anybody does CLEM on a DNA paint sample. So look, here again, we label the cells for LAMP1. And this is the signal of a single lysosome. This actually is an endosome because it's a late endosome because it's empty. The scale bar is about 50 nanometers. And you can see how the fluorescent signal perfectly colocalized, like correlates with the shape of these vesicles at the EM. And also that you can literally count the molecules. So each dot is one antibody. So DNA paint is also exceptionally good at giving you objective data, quantifiable, and something that other CLEM techniques are not. This instead is a lysosome, is labeled for the same marker, but you can see that in this case it's full of material inside. And so this tells us that this is probably a lysosome. And again, that's why it's important to always keep the EM together. And to conclude, since we are now at the nanofib, we couldn't show any 3D or volume data. And for this, we did it in collaboration with my old PI, Tom Kirkhausen at HMS, and Elliot, who's the scientist who performed the imaging of the FIBSEM and the segmentation of the images are going to look. And we took a piece of hippocampus, imaged it in STORM, so in super resolution, and then Elliot performed a volume of a section, of a small portion of that sample. So what you see here are like 3 consecutive sections of that area, like 1, 2, 3. Each one is about 250 by 370 microns. See? And then out of these sections, Elliot picked a small region for fibbing. And what you're seeing now is, if you remember that section that you saw before, that 1 micron thick sections, we are looking at from top to bottom in this, in the Z direction. So you have all your nuclei, the cell bodies, the axons. And then we took this sample and we labeled for VAMP1, that is a synaptic marker for inhibitory synapses. Then we chose these particular synapse, and Elliot was able to segmented at a resolution of 10 nanometer voxels. So we're able to look at the mitochondria, the active zone, and the vesicles of these particular synapses. So look at this. There are two active zones, one in front of each other, and there is like the vesicle cluster and the mitochondria here. And all of this has been done relatively seamlessly because you're only dealing with a small portion of tissue instead of having like a big volume to then look your fluorescence for. So the take-home message from this talk is that TOCLEM works in all or most kind of sample. For now, we didn't find a sample that didn't work on. It allows us to label for multiple macromolecules. It's compatible with several super resolution techniques, and also for volume EM analysis of the same sample. And thank you again to all our collaborators, but in particular to especially Abigail, that is an amazing director of the facility and allow all of these and makes everything possible, and also to Yoon, Peggy, and Manko, and David. So thank you.